So welcome everybody. So this is the second meeting of the Meet the Scientist webinar series. As a reminder, the Meet the Scientist webinar is an opportunity to meet different teams and learn about the exciting research that they are doing in Alzheimer's disease and also other dementias. Today is my great pleasure to host our colleagues from the Vanderbilt Memory and Alzheimer's Center. And I'm going to start by introducing Dr. Angela Jefferson. She's the director of the Vanderbilt Memory and Alzheimer's Center, and also the director of the NIA-funded Vanderbilt Alzheimer's Disease Research Center. She's a licensed neuropsychologist and a tenured professor and vice chair of scientific strategy and innovation in the Department of Neurology at Vanderbilt University Medical Center. Her research uses a cognitive neuroimaging, cardiac imaging, molecular biomarker data to study hemodynamics and pathogenesis and clinical manifestation of Alzheimer's disease, cerebral small vessel disease, and neurodegeneration. Please join me in welcoming Dr. Jefferson and her team at Vanderbilt. Thank you.
Thank you so much. I'm gonna share my screen, and I'm also gonna turn off my video since my Wi-Fi is shoddy this afternoon, so that hopefully this works. So thank you so much for the opportunity to share information about the Vanderbilt Memory and Alzheimer's Center. Before we jump in to some individual faculty research program descriptions, I want to just provide a brief background about our center and research and training environment for the audience. So our center's mission is to solve the complexities underlying the pathophysiology, early identification and treatment of Alzheimer's disease and related dementias, as most Alzheimer's Center programs are. We were established in 2012 in the Department of Neurology, but earlier this year in 2020, we transitioned into a freestanding institution-wide center, and we now serve as the hub for all clinical research and education activities in the ADRD space across the Vanderbilt campus. Our interdisciplinary faculty include 50 individuals from Vanderbilt University Medical Center, and Meharry Medical College, representing 19 departments that range from biochemistry and pharmacology to emergency medicine and health policy and everything in between. And our research emphasis of the center is basically our prevention and treatment strategy focuses on concomitant pathways of injury that commonly co-occur with core Alzheimer's pathology and contribute to the clinical manifestation of disease. Our center offers an interdisciplinary Alzheimer's T32 training program, and this campus-wide program was developed with leadership from neurology, biochemistry, psychiatry, and medicine, including Dr. Sanders, who you'll hear from later on this afternoon. Our training faculty includes over 30 faculty members, which also includes 3 Alzheimer's-focused K-24 faculty mentorship grants. And our trainees are quite interdisciplinary. We have PhD students in neuroscience, material science, and chemistry, and we have postdocs in neuroscience, chemical engineering, and chemistry. Our curriculum includes an interdisciplinary journal club that's hosted by yours truly, along with Dr. Matthew Schrag and Dr. Fiona Harrison, who you'll hear from today. We routinely host more than 40 attendees with about a dozen or more faculty consistently attending. We offer a grant workshop for trainees and faculty and a guest lecture series. And we host an annual Alzheimer's Disease Research Day that features faculty presentations, data blitz, keynote presentation, and a networking poster session for the entire campus. Our 2020 event is actually scheduled for later this week, and we have nearly 200 faculty, staff, and trainees registered to participate across our campus, which really, I think, highlights the robustness of our community. But collectively, I think these activities over the last few years have really fostered a rich intellectual neighborhood for us and a cohesive training and collaboration environment. Another important resource I want to describe briefly is the Vanderbilt Memory and Aging Project, which is known locally as VMAP. It was established in 2012 to delineate the complex link between cardiovascular health and brain health, including the risk of cognitive decline in Alzheimer's disease. And the original cohort, which was funded by an R01 grant that I had, includes a few hundred participants with and without cognitive impairment who were phenotyped in pretty rich detail over a 5-year period. And then recently, Dr. Catherine Gifford, who you'll hear from shortly, received an R01, and I received a R01 as well, a renewal of that R01, that in combination will support about $23 million of an expansion to follow the legacy cohort we established and enroll another 650 participants with identical phenotyping over time. And I just want to highlight the detailed sort of data types that we're collecting. So we collect demographics and clinical history. We have, I think, over 12,000 mils of blood stored right now. We have done GWAS and RNA-seq on these participants. We have detailed cardiac imaging, echo, cardiac MRI, ambulatory blood pressure monitoring, detailed cognitive testing. We collect a 65-minute neuroimaging protocol that has considerable structure and function variables, and we collect molecular biomarkers. Our biobank currently has about 10,000 mils of CSF in it from these participants. So quite a lot of data. In terms of the new areas of focus, because we know that pathology associated with Alzheimer's disease and dementia develops 20 to 25 years before clinical symptoms ever manifest, the expansion will enroll younger participants, age 50 and older, who are cognitively unimpaired. And to better understand disparities in Alzheimer's, we'll allocate significant resources to Black and African-American participant outreach, which will be led by a new dedicated community outreach and engagement manager who we hired in June. And finally, to elucidate the intersection of cardiovascular risk and Alzheimer's risk, we'll universally collect cerebrospinal fluid on most of the cohort for detailed in vivo molecular biomarker analyses. So about half of the original cohort has CSF, and then that'll be a more consistent requirement for the expanded cohort. Another thing that I want to mention is that this past August, we received an ADRC P20 grant. So Vanderbilt's one of 4 institutions nationally to receive a P20. And the leadership structure for this new Vanderbilt ADRC includes Associate Directors Dr. Consuelo Wilkins and Dr. Chuck Sanders, who you'll hear from shortly. The clinical core will be led by Dr. Paul Newhouse, and Dr. Catherine Gifford will serve as co-leader. And the biomarker core will be led by Dr. Timothy Hohman, and co-led by Dr. Bennett Landman. You'll hear from both of them as well. I want to briefly mention that aside from what I think are our fantastic people at Vanderbilt, Vanderbilt also has a really long-standing commitment and tradition to professional development of students, postdocs, and early career faculty. And I want to highlight just a few of these programs, which I think are quite unique to our environment. So first, our Biomedical Research Education Training Office, which we refer to locally as BRET, has this NIH-funded Career Development Aspire program for PhD students and postdocs. And this is a pretty intensive curriculum that offers opportunities for postdocs and trainees to explore, to do career exploration internships, the professional development workshops, and skill building workshops. And so there's basically curriculum almost every week for our trainees who can participate in this. And that includes academic-based directions as well as industry-based directions for career. We also have an NIH-funded initiative for maximizing student diversity. And this program has been funded for 20 years and works to increase the number of biomedical science PhDs awarded to students who are underrepresented in science. The program offers a supportive community of PhD student scholars, opportunities to cultivate leadership skills, networking opportunities, and professional development. And this program has supported over 100, and I can't even keep track of the number anymore, but an extensive number of students over the last few years. The Edge for Scholars is actually a national resource. Anyone can benefit from this, but it was founded at Vanderbilt and is a really unique web and social media platform that's curated around the needs of academic biomedical researchers. And it covers the full arc of one's career, but there is a special emphasis on the needs of early career investigators. So quite a few of their blogs and articles to sort of guide early career faculty. Vanderbilt also offers the Academic Pathways program, and this is a program that recruits and prepares postdoctoral fellows from diverse ethnic and racial backgrounds for competitive academic faculty careers. I sit on this, the advisory board for this program, and it's a really unique and remarkable program. We also have a Women on Track initiative that's focused on increasing the number of women on the tenure track and their rate of promotion by addressing gender-specific issues related to career development. And our very own Dr. Fiona Harrison previously served on their steering committee. And then last, we have the Vanderbilt Faculty Research Scholars Program. And this is an institutionally funded career development program for early career faculty investigators. So it's like a career development award, only it's institutionally financed. And it provides 75% protected time and salary for investigators. And it also funds research costs. And this is meant to be sort of like a mechanism to help support faculty who are applying for external K awards. So this is just a subset of some activities that we have on campus. There are literally many, many more that we could highlight, but I wanted just to mention these. And so I also want to mention that with so much growth and new grant funding comes increased training and faculty recruitment opportunities with our faculty and center in particular. And so each faculty member today will mention training opportunities that they're supporting in their individual laboratories or programs. But I do want to mention 2 faculty level opportunities for which we're actively recruiting. The first is an early career behavioral neurologist, and the second is an early career geriatrician. So if you're interested in these opportunities, check our news and events section on our website, which is here, vanderbiltalzheimers.com. And for the latest on training or job opportunities, you can also follow us on Twitter at vanderbiltalz, or you can follow my Twitter at AJ the Scientist. So I thank you very much for your attention. And now it's time for you to hear from our scientists. So we're going to start with Piper. Hello, let me stop sharing. There we go.
Hi, everyone. Let's see. Can everyone see my slide, my one slide?
Yes.
Oh, doesn't want to go into full screen mode. Oh, no, now it's crashed. Let's try one more time. Sorry about that. I've got another idea. Let's do this a different way. So if I drag this over that, can you guys see my slide now? Does that work?
It's, yep, that's perfect.
Here, I'll adjust it just a little bit so I can actually also see it too. There
now we can see your news bar on the right.
Yeah, there we go. There.
Perfect.
Okay, awesome. So yeah, thanks so much for having me today. I am Piper Below. I run the Below Lab at Vanderbilt. I'm a member of the Vanderbilt Genetics Institute. I'm an associate professor here. I've been at Vanderbilt for about 3 or 4 years, and my lab, among other things, studies computational genetics around Alzheimer's disease. So I'm a computational human geneticist by training. I work in statistical genomics. And one of the main questions that we have in my area is how can we better understand how genetic variation translates to effects that increase risk of disease, and namely Alzheimer's disease. So we know that genes and variants can have impacts on gene expression levels. And so there's a concept called genetically regulated expression, which we've sort of shorthand termed GREX. The advantage for analyzing diseases like Alzheimer's disease in the context of genetically expression is that it reduces noise from temporal and other environmental factors, and we're able to aggregate multiple variants effects around a single interpretable unit, a functional unit, in this case, a gene. And also it has the advantage of, instead of doing millions and millions and millions of tests as we would be doing if we tested every single genetic variant individually, we're testing just the overarching number of genes. And so we reduce our number of multiple tests significantly by using this approach. So we've aggregated data from many different genetic databases that were designed to study genetic risk factors for Alzheimer's disease in a large-scale data reanalysis project funded by an R01 from the NIA. And what we found so far is our preliminary results have identified 293 significant genes that are behaving in both tissue-specific and cross-tissue fashion. And 72 of these genes are actually novel AD genes that have been never reported in previous late onset Alzheimer's disease genome-wide association studies, and that exist more than 10 megabases away from any of the previously reported SNPs. So this is a little Manhattan plot showing you in colors, the location of these new hits. Notably, what we find is that the regions of the genome that are surrounding our known and novel gene hits that we're identifying through genetically regulated expression actually are highly, highly enriched for heritability, accounting for Alzheimer's disease risk. So in summary, we're able to increase the power of genetic discovery from common genomic variants with this approach, looking at genetically regulated expression. We've identified a large number of genes across a wide array of tissues, both what you might consider your canonical Alzheimer's disease tissues in the brain, as well as in cross-tissue analyses and in single tissue analyses in sort of non-canonical tissue types, which is a really important finding. We're continuing to aggregate these data sets and hone tissue expression profiles for Alzheimer's disease for improved characterization of genetically regulated expression in additional tissue types as this project goes on. So that's the main focus of our lab within the Alzheimer's disease space is to expand these kinds of analyses as we can learn more about gene expression levels that are pertinent for Alzheimer's disease risk.
Great, thank you so much. So next up is Dr. Gifford. Katie, do you want to share your slide with us?
Can everyone see that? Is that shared? Okay. So I'm Catherine Gifford. I'm a assistant professor and neuropsychologist at the Vanderbilt Memory and Alzheimer's Center. And my research focuses on refining early detection of Alzheimer's disease with a specific focus on using clinical metrics. So these would be things that are easily accessible, time and cost efficient, and highly, and ideally part of the clinical exam that's already going on. And the one focus of the project is knowing that there are various different drivers that lead to objective or subjective cognitive changes. So this can be Alzheimer's disease, but also neurodegeneration, cerebral vascular disease, or even mood symptoms. And here's just a chart from data from the Vanderbilt Memory and Aging Project that Angela talked about, highlighting some of those pathways. And the main, one of our real focuses is to create profiles or clinical profiles that would help us identify underlying pathways that might be driving some of these objective and subjective changes, and understanding how these pathways overlap, so that we can provide more tailored and targeted interventions as they become available, but also some prevention. We do this using cerebrospinal fluid biomarkers, also markers of hemodynamics, so both using brain MRI and cardiac imaging, as well as brain MRI metrics looking at micro and macrostructural changes. And I'm happy to take any questions. I have my email there as well too.
Great, thank you so much, Katie. Next we have Dr. Hohman. Tim, do you want to share your screen?
Hey everyone, actually I'm not going to share a slide. I thought I would mix it up a little bit and just
That's great.
go without a slide. So my name's Tim Hohman. As Angela mentioned, I'm the director of the Biomarker core here. I was trained as a neuroscientist and then did a postdoctoral fellowship in human genomics. So the focus of my team is really on how we can use multi-omic and multimodal data in a precision medicine approach in Alzheimer's disease. So we both focus on how we can stratify patients to identify the most at-risk patients, how we can integrate considerations around sex and gender and race into our models to better understand risk and resilience in Alzheimer's disease. And then a big focus is looking for genomic contributors to resilience in Alzheimer's disease. So that's what I'll talk about for just a minute or two. So one of the things that we're really interested in are people who have all of the neuropathology of Alzheimer's disease, but don't show clinical symptoms. And we use neuroimaging and biomarker and autopsy data to define those individuals. And then we use genomic techniques to identify genes and molecular factors that may contribute to that resilience. And our hope is to use those markers to move them towards therapeutics. So we have collaborations with mouse model researchers at the Jackson Laboratories. We have collaborators here working on proteomics. You'll hear from Renã Robinson shortly, Dr. Matthew Schrag and Dr. Ethan Lippmann we work very closely with as well. And then we have some external collaborators where we're running large-scale proteomic analyses within the Religious Order Study and Rush Memory and Aging Project. So our goal is really not just to identify genetic effects, but also to translate those into therapeutic targets using other multi-omic approaches. So happy to take any questions, but that's my group.
Great, thank you so much, Tim. Next we have Dr. Lippmann. Ethan, are you going to share a slide with us?
Yeah, I'll stick to the traditional route and share a slide. Hang on, I have a million slides open on my computer. I won't go into presenter mode. So hi everyone, I'm Ethan Lippmann. I'm an assistant professor here at Vanderbilt in the School of Engineering. And The mission of my lab is pretty comprehensive, to model, understand, and hopefully one day treat neurodegenerative disease by focusing on blood-brain barrier and neurovascular function. And our general strategy is to combine biomolecular and biomedical engineering principles with molecular biology techniques to facilitate very comprehensive in vitro and in vivo studies. So I'm an engineer by training. All of my degrees are in chemical engineering, but all of my research has always been in stem cells and neuroscience. And so, I kind of bring both perspectives to the team in that sense. We're a very broad lab. On the engineering side, we build complex neurovascular tissue models from stem cell sources and primary tissue. And then we use engineering strategies, molecular recognition strategies to enhance various applications that we're pursuing in molecular targeting, biosensing, and drug delivery. And then more on the molecular biology side of things, we use our rudimentary cell models to dissect neurovascular function and signaling pathways. In vitro, we use human tissue and our cell models to investigate mechanisms of neurodegeneration and neurovascular disease. And then we do rigorous validation in vivo models, as well as our more complex tissue models. And we utilize a variety of engineering and molecular biology techniques. So we do a lot of high throughput CRISPR screening in cell-based models. We do biomolecular engineering. So we used in vitro selection and directed evolution to generate targeting modules for delivering drugs to the brain, for instance. The engineering side of things, we build a lot of our own devices. So this is a spinning bioreactor. These are microfluidic devices. This is a little widget that can change media on every well of a 24-well plate simultaneously. And then we also make our own biomaterials to enhance our tissue models. So these are biomaterials that promote the maturation of neural cells in 3D culture, generate vascular sprouts, things like that. So I have a really large and diverse lab. I think I have 16 trainees right now, spanning engineering all the way to molecular biology. So we have a lot of expertise in the lab to do a lot of really diverse activities, and that's definitely the way that I like it.
Thanks, Ethan. Next is Dr. Sanders. Chuck, do you have a slide you want to share with us? Chuck, you're mute.
Good afternoon, everyone. Sorry about that. So I'm Chuck Sanders. I'm in the Department of Biochemistry here at the university. And I suppose I'm probably one of the more basic scientists in the entire P20. And so we've been doing Alzheimer's related projects for about 15 years. And our main focus has been on the protein that's the immediate precursor to amyloid beta. And it's actually a membrane protein called C99. And so a number of years ago, we determined the structure of this protein in model membranes. And we then also titrated the protein with cholesterol. And what we found is that C99 forms a stoichiometric complex with cholesterol with a KD that is physiologically relevant. And so there's about 3,000 papers on cholesterol Alzheimer's disease relationships. So we think that this observation may link the amyloidogenic pathway with the role of cholesterol in Alzheimer's disease. And this is actually a good point of entry for my lab into this project team, because as you've already seen, a number of labs have interests in neurovascular risk factors for Alzheimer's disease, one of which obviously would be elevated cholesterol. And so in the recent years, we've been trying to figure out exactly what is the relationship between cholesterol and the amyloidogenic pathway, and how might this promote Alzheimer's disease. And so we're continuing these studies after a number of years of going after them. The other thing we're working on, the other focus that you heard from some of the other investigators, is looking for new risk factors for Alzheimer's disease. And so we're keeping our eye on these lists of risk factors. in the hopes of finding the perfect protein to study that would allow us to use our sort of biophysical and biochemical techniques to illuminate exactly what are the mechanisms by which one of these new risk factor proteins does in fact promote the disease. And we've been talking a lot with Matt Schrag, and we will continue to talk to him and others on the team. And we'll probably eventually start a second project at some point based on pursuing one of those risk factors. So thanks very much.
Wonderful. Next up is Dr. Schrag. Matthew, do you have a slide to share with us?
Yes, I do. I didn't realize we could go rogue, or I might have tried that. Let's see if this works. All right. Can you guys see that okay?
Yep.
Great. So I'm Matthew Schrag. I'm a neurologist with a clinical interest in cerebral amyloid angiopathy, which is a vascular disease where beta amyloid is deposited in the blood vessels, and it's related to cognitive impairment, and it's part of the complexity of Alzheimer's disease that some patients have mixed pathologies, and this is one of those mixed pathologies. So we started this laboratory about 4 years ago with the intention of looking at two big projects, and we're currently recruiting a postdoc for project 1, and we'll be recruiting for project 2 in the next couple of months. So if this piques your interest, please get a hold of us. Project number 1 is looking at lysosome function around neurons, and we've known for 20 years or so that lysosomes accumulate in dystrophic neurites, so these distended axon segments around beta amyloid plaque. So here we've stained a beta amyloid plaque, and you can see these large accumulations within the axons that run past it. This is both in a mouse model of Alzheimer's disease, but also in human tissue. There are these large collections of lysosomes that don't belong there. And in addition to being related to beta amyloid pathology, we see little bits of tau accumulating in these as well. And we've magnified one that we've taken in cross-section to show you that these neuropil threads, which are a pre-tangled tau aggregate, form right in the middle of these dystrophic neurites and are surrounded by lysosomes, which are dysfunctional. So we've been leveraging some novel in vitro methods to try to get at this. One way to put that structure, that dystrophic neurite structure, in a dish is to do organotypic brain cultures, which we have thoroughly optimized over the last couple of years to get to function in adult tissue. Because the brain has to be aged to a certain point before you see these dystrophic neurites. And we're starting to get some really phenomenal results from that, including what we're seeing down here in the lower row, where we can see lysosomes stained with live imaging probes. And what we see is that neurons avidly take up these probes, but the dystrophic neurites seem to fail to acidify. These are pH sensitive probes, and it shows that the lysosomes and cell bodies here are much, much brighter than in the dystrophic neurites. Moreover, when we expose the cultures to baflomycin, which poisons the vacuolar ATPase, which is responsible for acidifying lysosomes, we can make that cellular signal completely go away, but the dystrophic neurites don't respond. So we're looking at trying to understand the molecular basis for why there appears to be a problem with acidifying lysosomes around beta amyloid plaques and what the relationship with that and tau pathology is. The second project is the one we've been at a little bit longer. We've been doing clarity to try to take human tissue to an optically clear state, and we have optimized the living daylights out of this to get human brain tissue as clear as glass. That piece of brain tissue sitting in that dish right there is about 5 millimeters thick. You should not be able to see through it. And when we put it under a microscope, even a conventional light microscope, we get this beautiful look at how the blood vessels are shaped. Normal blood vessels have a nice, smooth branching pattern and thin wall. And with cerebral amyloid angiopathy, when beta amyloid is deposited in the blood vessels, the wall thickens and becomes lumpy and bumpy. And as the disease progresses, we see these alternating strictures and dilations as the blood vessel degenerates and ultimately results in hemorrhage. We can take this type of clear tissue and place it on a light sheet microscope, which allows us to rapidly obtain three-dimensional images. And I've given you a quick example of what that can look like in the lower frame. Beta amyloid is stained in red, and the blood vessel endothelium is stained in green, a yellowish green. And we've done a real modest reformat here because you'll crash Zoom if you do anything bigger than this, but you can get really beautiful three-dimensional renderings of the vascular tree. And we're using this to model microvascular degeneration as a network to see how beta amyloid affects the microvascular network. So again, we'll be recruiting postdocs for both of these projects, and we'd love to see some new talent around here.
Thank you so much, Matthew. Tim is making jokes about how you win the award for best slides, best graphics. So the next couple are videos, and I am praying that this works well. I'm gonna turn my video off and then play the video for y'all. So we'll see how this turns out. Okay, can y'all see that?
Yeah, we could.
Okay, you can see it?
No.
Not, you can't see it now?
No.
Okay, hang on. Let me try this again. All right, let me just go to the next one. I'll do a different one. Fiona, do you want to do this for me since I'm having trouble? Do you mind handling it? Just start with Bennett's.
I can try. Okay, I have that. Let's get the.
If not, we're going to have to do their intros for them, which should be pretty fun.
They just
do it.
So this is
Hi everyone, I'm Renã Robinson. I'm an associate professor of chemistry and neurology. And here you can see I have a very diverse group of young scientists that range from folks with chemistry background and experience to individuals with neuroscience backgrounds, as well as medicine, health, and society in a variety. So we're a highly interdisciplinary team of researchers, and we have undergraduate researchers all the way up to senior research scientists that are part of our team. So in our lab, we're very much interested in the fact that there are racial disparities that exist in Alzheimer's disease, specifically in the Black and African American communities. There's 2 to 3 times higher incidence of Alzheimer's disease. And what you see here in this infographic is that African Americans are also greatly underrepresented in basic research studies.
We can't hear when you mute yourself, Fiona.
And the basis of these disparities, how these disparities come to fruition, and how we might be able to use these technologies to ensure that the findings that we as a community discover in Alzheimer's disease are going to be helpful for everyone. So my lab has expertise in proteomics. We also do some lipidomics research. And while we focus on racial disparities, we have a variety of other projects in the labs that span looking at the basic biology of aging to also understanding racial disparities in other areas. Our expertise lies specifically in using mass spectrometry to do our analysis of proteins. And so we have a number of state-of-the-art mass spectrometry instruments in our lab and very high throughput sample processing procedures that allow us to look at samples that can come from clinical patients, as well as looking at samples that come from various tissues within animal model systems. And so ultimately, the heart of what we do is we measure the masses of proteins and peptides or lipids, and we use information about the quantities of those individual proteins to help us understand more about disease. If you have further questions about the research that we're doing in my lab, I'd be happy to answer that during today's session or offline via email.
So Fiona, I think I can get the next one worked out. Let me just share and let's see how this works.
Hello, thank you, Angela. My name is Bennett Landman. I'm from Vanderbilt in electrical engineering, but I work closely with the VMAX Center on a variety of projects. Our main interest is, as this picture shows, the diffusion imaging of the brain. How can we understand the wiring, the dyswiring? How do we understand the connection structurally from one part of the brain to another? To do this, we bring forward a lot of different types of technologies, but the key one is called diffusion-weighted imaging. And this uses very powerful magnets in the form of a magnetic resonance imager to weight how the pictures come out of the brain, not by the type of tissue, white matter, gray matter, lipids, solids, water, that kind of thing, but rather how the tissue is oriented on a micrometer scale. We can literally see the water moving down the micron tubules of axons on a millimeter scale. And from all of those little tiny bits of water, we piece together these incredibly intricate pictures of the brain. Now, we have technologies to do this. We have a lot of experience in this, but unfortunately, it's not always as accurate as we would like. And that's where our research comes in. How do we take these images that are beautiful, have been shown to be correlated with Alzheimer's disease, aging, development, and how do we make them more quantitative? To do this, we bring forward a couple of ideas. First is in the image acquisition. How can we work with the biophysicists to get more reliable acquisitions? How can we work with the anatomists and the ontologists to understand what should be there, what's an error, what's not? And finally, where our expertise really comes in is in the image analysis. How do we take those pictures, the pixels, the voxels, the little bits of information that are measured, and bring them together into a quantitative story? To do this, we often stick in phantoms or chunks of water or bundles of fibers to see what's actually happening versus the physical thing we put in there. We look at quantitative modeling on a very fine scale and ask, is this the right estimator for what's going on? And can we link this with the structure? Can we link this with other imaging modalities that tend to fail? And then finally, because we're in machine learning and I'm at computer science appointments, how do we bring machine learning into this? How do we use AI to help us do all of these things better, from optimizing the acquisition, to interpreting anatomy, and in looking at the images as pictures and as quantitative beings? So hopefully you'll be interested in joining me. My email address is right above me at the top of the screen. Feel free to shoot me an email, and I'd love to chat. Thank you very much, and back to you, Angela.
Okay, great. So now we're gonna.
When we think of Alzheimer's disease, we typically think of.
Let me do a proper introduction here. Next, we're gonna hear from Dr. Fiona Harrison. Here we go.
We typically think of older patients struggling with the later stages of illness when cognitive, emotional, and behavioral symptoms have a major impact on the lives of sufferers. In my laboratory, we are much more interested in the very early stages of the disease, when changes in the brain are more subtle. We study dietary and environmental factors that can accelerate the pathogenesis of disease and lower the age of onset of detectable behavioral change. We believe that a pathway that can be accelerated can also be slowed down. And so we try to identify those factors and pathways that can be manipulated to be protective against future decline. In particular, we focus on vitamin C deficiency and excess manganese exposure and their effects on dysregulation of the dopaminergic and glutamatergic neurotransmitter system. Both of these molecules have several routes of action, including acting as enzyme cofactors and altering the redox balance of the brain. We use a variety of methods with a strong focus on in vivo testing of behavior and long-term EEG measures that support our findings with a wide range of wet lab techniques to understand the roles of vitamin C and manganese on the neuropathology of Alzheimer's disease. In two recent findings, we use gulo knockout mice that are, like humans, incapable of synthesizing their own vitamin C. The current graduate student in the lab, David Consoli, showed that in 12-month-old genetically modified mice, chronic low vitamin C, or ascorbic acid, was as disruptive to dopamine release and reuptake in the nucleus accumbens as carrying APP and PSAM1 mutations, independent of the related amyloid and oxidative stress pathology. In a study from 2018, a team of graduate students, postdocs, and several collaborators demonstrated that vitamin C deficiency increases the likelihood of observing spike discharges under baseline EEG conditions, and increases overall number of tonic-clonic seizures experienced following a mild kynic acid challenge. Perhaps even more interestingly, some newer preliminary data showed in very old 19-month-old gulo APP-PSAM1 mice, depletion of vitamin C in the brain over the course of 5 weeks led to a decreasing theta ratio. And one interpretation of these data is that it reflects changes in wakeful attentiveness. Although chronic mild deficiency like this may not be causal for disease, it can still directly impact some of the behavioral symptoms observed, including activity and motivated behaviors, or may modify effectiveness of pharmacological interventions. Findings like these are particularly important given that the majority of preclinical work is conducted in standard lab rodent models in which nutritional deficiencies or even dietary variation are rare. Both dopamine dysregulation and subclinical seizures are acknowledged but understudied areas of interest in Alzheimer's disease research and may prove important in identifying new routes for therapeutic intervention. In our future work, we plan to incorporate many of these same approaches and a few new ones to also probe the roles for manganese neurotoxicity and progression of disease, as well as continuing our work with the neurobiology of vitamin C.
Great, thanks so much, Fiona. So last, but hopefully not least, I'm gonna screen share and go through a little bit of my own work. There we go. There we go. Okay. So I am a neuropsychologist, clinical neuropsychologist, and a professor of neurology. And my research program has a few interrelated areas of focus. So first, I'm interested in the impact of cardiovascular aging on Alzheimer's disease and cerebrovascular disease, and really cerebral small vessel disease. And this cartoon just illustrates the cardiovascular aging from the heart through the aorta up to the carotid and into the cerebral macrocirculation and microcirculation, all of which we're quantifying in rich detail in our local VMAP cohort that I described earlier. We found that subtle age-related changes in cardiovascular function and arterial stiffness affect cerebral blood flow, especially in the temporal lobes, as well as cognitive trajectory and risk for dementia. A second area of focus within my program is delineating the etiology and clinical consequences of cerebral small vessel disease. In VMAP, we're quantifying cerebrovascular structure and function in rich detail so we can ask questions like, do certain small vessel disease markers have a greater impact on cognition than others? And the answer is we believe yes, that perivascular spaces, as illustrated here via electron microscopy, have meaningful clinical consequences. I'm also interested in the intersection of cerebral small vessel disease, Alzheimer's disease, and concomitant pathways. And while mentioned that while most of our work has been in human observational cohorts like VMAP so far, we're actively expanding that work into animal model systems, cell model systems, and omics in collaboration with many of the folks you've heard from today, Drs. Hohman, Harrison, Robinson, Lippmann, and Schrag. I want to mention that we have training opportunities for PhD students, MD PhD students, and postdoctoral fellows, as well as opportunities within our T32 program and the faculty recruitment opportunities that I described earlier. So for more information, you can check out our website at vanderbiltmemory.com that has a news and events section where we often post opportunities. You can email me at angela.jefferson@vumc.org. And the team has finally got me on Twitter, so now I'm learning how to tweet. And so you can follow me on Twitter for training opportunities that we'll post there as well. So thanks very much for your attention.
Thank you, everybody. This is very impressive. I learned so much, and I really appreciate you taking the time to share with all of us everything that you're doing. There were some questions that I know they already answered one of them, but they will be important to go through the questions, especially the ones that I have opening for postdoc and junior faculty positions, since many of the attendees are actually early career. So I don't know, Angela, if you want to.
Yeah, so what I would encourage folks to do, we have a lot of different faculty development programs. I highlighted just a couple opportunities that we have, but there are many more. And so if people are interested in faculty opportunities, they can email any of our faculty, including me, with their CV and their interest. And then basically as a freestanding center, we partner with different department chairs to facilitate recruitments. And so we try to find the right home in terms of department for where that person might build their career. And so we can sort of direct them to the right leadership for potential recruitment. So
perfect, thank you.
And I think I saw also that there was a question about our amyloid, like our PET capacity. One of the things that Fiona and I had talked about last night was wanting to highlight some of the important resources, core infrastructure that we have at Vanderbilt. One sort of unique thing about Vanderbilt, I think, is that there's been a massive investment over the years in core resources and shared resources, so that rather than having to have really fancy microscopes and animal behavior tools within your laboratory, they're shared resources where you can pay a subsidized rate to access them. And one of those important resources that we've utilized quite a bit is the Vanderbilt University Institute for Imaging Science. In that institute, we have it's a research dedicated institute. And so we have 2 research 3T MRI scanners, a 7T MRI scanner, multiple animal scanners, and a PET CT scanner with a cyclotron. And I think Tim wanted to answer the question specifically about kind of our current PET capacity. And so Tim, do you want to add to that?
No, what you just said is what I was going to say. I'm typing some additional details, but yep.
Okay. And so just to add to that, I'll say that we can we have the capacity to do PIB and obviously any F18 tracer as well. So.
Perfect. And somebody actually asked me a question, and I think that is important to share with the group. So, yeah, somebody was asking about Vanderbilt, and also more like in terms of how is the institution, you know, like the environment, like even beyond the center. So I don't know if you can share a little bit more about that, and even like sharing something about the city, you know, like anything that the people can be excited about.
Yeah, so I would say that Vanderbilt is rapidly growing. It is, I came in 2012 and it's so different now. It's one of the most rapidly growing cities, I think, in the country. It's also a really fun city. We have our professional sports teams are finally winning a bit, which is after leaving Boston, that was hard to come from a big sports town. But yeah, it's a great community. Does anyone else want to speak up? It's like sort of chime in on what they like about. Hi, Renee. Do you want to chime in on what, or Fiona, you turned off your mic first. Do you want to chime in on what you?
You know, when I'm trying to sell Nashville to people, I mean, for the most part, it sells itself. It's just got a bit of something for everyone. So if you like to get out in nature and hike, we have all these national parks, kind of little parks in the city, really accessible hiking, climbing, all that stuff just outside the city. If you're a city person, we have all the things that you need, you know, and shopping. And of course we have music venues of every description, which, you know, hopefully by next year will be, you know, open again and we can all go out and do that. So it's got everything that you need from a big city, but you don't feel like you're living in New York or, you know, Boston or somewhere where it's just a lot busier. It's a really homely place, and it's the South. And Vanderbilt is very much like the whole of Nashville in that everyone is just so friendly and collaborative. I think that's what we hear is the difference when people come here. They just really like the way people collaborate across departments, across centers, within centers, and it's just helpful and friendly. So I came for 2 years, and that was in 2004. And, you know, I maintain that I made the right decision by just staying.
Yes, it's very affordable. Renee, did you want to add something?
No, I'll just concur with what Fiona said. I mean, I think I came from Pittsburgh here to the city, and pretty much everything that I needed and looked for as an individual. I think for individuals coming from diverse groups, there is a lot of that here in the city. And so, you know, we have international markets, international festivals, just kind of everything that can kind of cater to everybody. If you have family, there's lots of family friendly things to do here in the city. And the weather's good. And yes, it is the South, but Nashville is the progressive part of the South. So I'll put that out there.
Matthew, you came from the West Coast. Do you want to say something about the West Coast trend? I know you hit East Coast before you came to Nashville, but you're a West Coast native.
This is a great place to live. You know, Sarah and I really looked nationwide when we were looking for a faculty position, and we're blown away. You know, it's one of the few places where I felt that my wife genuinely loved the city and I genuinely loved the university. And putting those two pieces together is sometimes really hard. This is a world-class university, and across the spectrum, there's just an incredible amount of resources, things to do to keep it really interesting and stimulating. And this city is outstanding. I can tell you, my wife and I are more rurally minded. We live 25 minutes outside of town on a horse farm, and I get right into town to do big city kind of work. So you can really make any mishmash of lifestyle work here. And I will say one other thing. I'm 4 years into a faculty position, and this is such a nurturing place to be a junior faculty. When I got here, I was told that if I took a good shot at this faculty development grant, I'd probably get it, and they were right. And it kept the pressure of getting that K award a little lighter. It meant I had time and I had resources to get started. And then the K award came much more naturally. And now I'm writing my first R01, and I don't feel like there's the pressure to survive that there is in some places. So it's just a great place to get your start.
That's a great point. Tim may correct me on the sort of actual statistic, but I'll say that we have a phenomenal K to R01 conversion rate. So Tim, do you want to highlight the statistics? Because I know you remember that stuff much better than I do.
Yeah, it's a really, so Angela brought up one of the programs that we have here, the VFRS program, but we also have a lot of programs that support the development of those external applications. So the K rate is very high, but the really impressive number is the K to R01 transition. That rate is between 70 and 80% at Vanderbilt. So really high number. And the reason is because what Matthew's saying is right. That's a really supportive environment. We've all experienced that. And then there's also really rich research communities here. Like I don't know many other places where, you know, you're seeing all of us in this presentation today, but we really do all see each other every month, and we really do all work together. It's not like a theoretical thing. That's something that really happens here. And I think that sort of supportive infrastructure where we're all, you know, bringing our grants to one another, talking through them, getting feedback, getting better at grant writing and getting better at science, I think is a pretty unique thing that Vanderbilt offers.
Yeah, so there's a program where through the EDGE for scholars that I highlighted, they started EDGE here, and the idea was that they provided mock peer reviews of trainee like K award and R01 grants. They record the actual review with the experts from around campus who serve on study sections, and they actually critique it just like a study section does. And so there's a lot of sort of formal things in place to help early career faculty write that K award and then get their first R01 funded successfully. Chuck, did you want to add something? I saw you take yourself off mute. I didn't know if you wanted to also say something.
Well, I was just going to say something about the graduate programs. As you brought up earlier, they really are remarkable programs in that they're designed to be very nurturing for the students. They're designed to promote diversity. We're among the highest producers of PhD minority students in the country. And a lot of emphasis on sort of non-traditional career paths, as well as the sort of, faculty or industry routes. And so it's just wonderful opportunities here. I think we most folks really sort of like are doing a job to be here.
Yeah, I think the IMSB, the Initiative to Maximize Student Diversity, that program has that formal curriculum to help with leadership skill development and mentoring, but it also supports the various trainees in that program engage in science outreach as well. And so it's really sort of like local, their community here on campus, but also the community, the scientific community across the educational systems within the Nashville area. So really neat stuff.
Perfect. And there was a question just to go over where can they find more information? I don't know, just to remind people about the website or where to get more information about the center.
Yeah, so the website's a good source for sure. And you can always email us and we can send links to the various programs of interest depending on what level people are applying to graduate school, if they're looking for postdocs, if they're looking for faculty positions, we can sort of direct them to the relevant resources for sure. It looks like there's one other question about what main factor drew us to Vanderbilt for science for training. So Tim, Fiona, and Katie, and Matthew all came as part of in a training capacity and then joined the faculty. I don't know if you want to answer that question.
Yeah, I can answer real quickly. Just that, I was really interested in an interdisciplinary environment. I wanted to be trained in genomics and still have my hands in imaging, and the imaging resources here are extraordinary and the genomic resources here are extraordinary. So it was kind of a unique opportunity to do that. And then as I came in, Dr. Jefferson was coming in too and setting up the Alzheimer's Center. So it just ended up working out really well that I kind of fit a niche center when I got here. But that's what drew me there then, and that's still true today, that the interdisciplinary expertise and ability to be trained across disciplines is pretty unique.
Yeah, I had a very similar story. I was hired as a behaviorist. That was my training, a rodent behaviorist, to fill a very specific role as part of an R01 project. And then when that when that investigator left, I moved to a new lab and got an entirely different set of training in molecular biology and a whole lot of other techniques. And I now have a molecular biology research lab, and I run the behavior core here. So I was able to use my basic training, but a whole lot of new techniques. And I would say I'm still learning techniques all the time through collaborations. And so that's really worked out well. I feel like a trainee half the time myself. So, but that was why I came, because I was just hired to do the one thing I knew how to do really well, but then I was able to leverage that to really expand my capabilities once I was here. Yeah, that's so true. I think the collaborative nature is really like one of the most fantastic things about the community that we have. I mean, many of the folks that are on this webinar have been in my backyard in the last 4 weeks having socially distanced cocktails when the weather's been nice. We spend time together, we do happy hours together. And there's always, everyone's always willing to lend a hand and help each other out to sort of, if they can't sort of provide the resource or the need, they'll find someone else on campus to help you. And so it is truly a really great community.
Okay, so I think that we are out of time, but thank you so much, everybody, for joining. And also thank you, the Vanderbilt team, for being with us today. And congratulations, by the way, in the new center. I think that is very exciting. And I think that's, yeah, big congrats for that. Thank you for taking the time. And just as a reminder, so the next webinar series for Meet the Scientists will be in December 3rd to meet the Healthy Brain Project team, that is our colleagues from Australia. Okay, so thank you very much and have a good day.
Thanks for coordinating it, Kel. Talk to you later.
Thanks so much.